Good morning, everyone, and thanks to the organizing committee members for this opportunity. My presentation will be a brief overview on the role of molecular pathways imbalances and their potential link with Alzheimer disease. As we know, Alzheimer disease is the most common neurodegenerative disorder worldwide affecting the elderly. About 10% of people of age 65 and older suffer from AD. Epidemiologic data in the last decade highlighted a sharp increase of AD incidence, and the current AD trend is alarming. It was estimated that over 150 million of people will develop AD by 2050. But what we know about Alzheimer's disease? Despite the years of research, our knowledge on AD pathophysiological mechanisms are not well understood. We know that there is a genetic component runs in a family and drastically increase the risk of developing AD. Part of this genetic background was identified into 3 genes that are APP, PCN1, and PCN2 that are involved, as you know, in the cleavage and the production of amyloid. Subject carried this alteration at the level of this gene showed an autosomic dominant inheritance. This part is well described in the literature. However, these cases only represent 5% of all AD cases. The remaining 95% background is still obscure. Of this selection of cases, which are also known as sporadic AD, sporadic Alzheimer, the best correlation found is represented by ApoE, and in particular by its E4 allele, which increases the risk of developing dementia 2 to 3-fold if it's present in heterozygous condition, and up to 12-fold if it's present in homozygous condition. But even if the risk is relatively high, the homozygous condition for E4 allele is not necessarily neither sufficient to develop AD. More elements likely can participate in order to trigger AD. So this is what we are searching for. We are searching for the 90% background for AD. But where can we start from? We can start from what we know. And we know that many studies, in particular many PVAS, have found various associations between genes that increase the risk to develop Alzheimer. But we know also that these results are controversial. And overall, the additional risk given by the presence of such variants is low. Probably we can explain it taking in consideration that each protein interacts with and influences many other elements in a synergical orchestra that regulates an organism. And we know that we are a really complex organism. As such, a single alteration propagates indirectly its effect, its interactors following pathways and molecular cascade. We think that rather than single genes, a better approach would be investigating AD as an event related to alteration affecting entire biological pathways. Another thing that we know is that in AD cases, in all AD cases, we have an alteration of beta-amyloid homeostasis with accumulation in beta-amyloid plaques, as well as the development of neurofibrillary tangles, which ultimately is led to neuronal loss. So we have thought to combine what we know. And so we have crossed the significant genes obtained from literature with the amyloid and tau pathways genes. In our hypothesis, whatever association gene found should be ultimately linked with the classical cascade, and finding the interacting elements, gene product, acting as a bridge and identifying the altered pathways involved in AD pathogenesis would, without doubt, help to better understand and characterize AD, possibly even finding AD subtypes. So with this cross, we have obtained a really big pathway, a really big group of pathway. So through pathway analysis and the use of bioinformatics tools, we have obtained over than 200 or 300, I think, different pathways, significant and important pathways related to AD. Subsequently, taking into consideration the p-value obtained and the literature data, we focused only on 10 pathways. That briefly I will show you. The first, just a moment, I want to move. Yes, oh, no, I can't. Okay, don't worry. The first one of them is, I want to move this one. No, can't. Sorry. Okay. The first one of them is oxidative stress processor pathway. And increased oxidative stress status is commonly observed in AD brains. A common feature of AD brains, in fact, is that they show an increased presence of reactive oxidant species compared to control brain slice. Moreover, the presence of beta-amyloid facilitates oxidative stress induction and represents a source of radical oxygen and nitrogen species that are prodromal factors associated to AD. Moreover, oxidative stress promotes tau phosphorylation, downregulates choline recycling from the synapses process, leading to HGH deficiency that are related to AD. This is directly related to the inflammation. Inflammation that we know plays a key role in increasing the severity of the disease by exacerbating beta-amyloid and tau nefarious effects. The main players behind the neuroinflammation and immune process are the non-neuronal cells that populate the brain, such as microglia, astrocytes, and oligodendrocytes. It is interesting to note that genome-wide association studies indicate that several genes related with an increased AD risk regulate the glia clearance of misfolded proteins and inflammatory reaction. Moreover, the presence of beta-amyloid stimulates microglia towards the chronization of a pro-inflammatory state by activation of a particular inflammatory cascade, that is the NFKB cascade. So we have the release of pro-inflammatory cytokines, release of proteolytic enzymes, and the production of free radicals, nitric oxide, and neurotoxic substances which can kill our neurons. Another process or pathway, if you prefer, is that related neurodevelopment and neurotransmission. Physiologically, these processes consist in the proliferation, differentiation, and maturation of a neuronal stem cell, and the modulation of their interaction through synapses and neurotransmission-related process. The synaptic pruning pathway becomes aberrantly upregulated in the first stage of AD. This aberrant activation, which leads to synaptic loss, seems to be triggered by beta-amyloid through the PAX1 ryanodine receptor function other than several inflammatory signals. For example, PAX1 is involved in the modulation of neurotransmission, neurogenesis, and synaptic plasticity, and the increased level contribute to neuronal death. Regarding neurotransmission, several reports have indicated a significant reduction of serotonin, acetylcholine, dopamine, and norepinephrine levels, as well as the receptor in AD brain. In fact, and in particular, in AD subject, loss of serotonin results in depression, anxiety, and agitation. Dysregulation of dopamine release leads to reward-mediated memory formation deficit, and a low level of norepinephrine impairs spatial memory function. In particular, cholinergic transmission and glutamatergic transmission abnormalities were pointed as one of the principal causes of cognitive deterioration in AD. For example, alteration of cholinergic transmission causes the accumulation of beta amyloid that degenerates basal forebrain in cholinergic neurons, and so on. A particular and interesting pathway that we have taken in consideration is glucose level and insulin pathway. For we know that alteration of processes related to energy production are distributed in AD brains. Constant high level of glycemia increase risk of developing AD. High glucose condition increases amyloid beta production by inhibiting APP degradation, and it causes an increase of insulin. In particular, insulin-related cascade has been the focus of numerous studies on AD. It was evidenced that the beta amyloid can interact with the insulin receptor, causing their internalization and thus inhibiting their function. This in turn causes a feedback which ultimately leads to insulin accumulation in the brain microenvironment. It has been demonstrated that the insulin accumulation triggers an inflammatory response since it is able to recruit different inflammatory cytokines. Of note, autophagy process that normally is an intracellular process mediated by vesicle and lysosome that consists of several sequential steps, which ultimately leads to the degradation of damaged or misfolded protein and dysfunctional organelles, thereby sustaining cellular homeostasis. Its impairment seems to be strongly linked with the exacerbation of AD pathologic because physiologically is really important for neurons and plays a defensive role in the nervous system. In AD brains, neurons, in fact, are rich in autophagic vacuoles were shown to be enriched in APP, gamma secretase components, PC1 and nicotine, which are required to generate the beta amyloid. And impaired autophagy lead to the accumulation and release of this material in the extracellular space, promoting beta amyloid plaques formation. From a molecular point of view, autophagy is mainly regulated according to the physiological condition of cell through several elements. For example, that was present in this slide. Interesting to note that the alteration of these elements have been correlated to AD, evidencing the existing link between the disease and autophagy. If we take in consideration the vascular pathway, we know that in patients with Alzheimer disease, cerebrovascular abnormalities are a common comorbidity. This may contribute to the onset of cognitive impairment and dementia. Altered cerebral blood flow and pressure at the level of the brain induces vascular dysfunction. These events are injurious to normal brain function that would result in disturbed homeostasis, but also in blood-brain barrier damage and microfracture in cerebral vessels, with increased risk of neuroinflammation and loss production. Moreover, are linked to the formation of beta amyloid plaque. All these conditions upregulate a particular signaling cascade that is a notch signaling cascade that has an essential role in vascular development and angiogenesis in brain through the modulation of a receptor that is PEGFR2 receptor. It has been observed that the chronic activation of NOTCH1 cascade negatively impacts the brain microenvironment, in particular the delicate connection of the brain with the cardiovascular system. Last but not least, the signal transduction process. In particular, alteration in PKC signaling, Wnt signaling pathway, and role of calcium. We know that protein kinase C is present in 3 different isoforms. We have a calcium-dependent PKC called the CPKC. We have a calcium-independent isoform MPKC, and we have a typical isoform APKC. All 3 isoforms are involved in several neural processes, including the ones related to cognitive function. In particular, the CPKC and MPKC isoforms could have impact on synaptic formation and plasticity, especially memory, organization, or dendritic loss, while APKC is involved in long-term memory. A deficiency in PKC isoform signaling is thought to be involved in a disease correlated with the tau hypophosphorylation and alpha secretase activity. Regarding Wnt signaling pathway, this plays a crucial role in the central nervous system and in memory creation and maintenance. Wnt impairments downregulates beta-catenin function, which in turn upregulates GSK3 beta, one of the main kinases involved in tau hyperphosphorylation. And role of calcium. We know that cellular calcium is a key ion involved in the regulation of several processes in neurons. The physiological calcium influx upregulates alpha secretase expression. Impairment of this process favors the formation of beta amyloid, and so the accumulation of plaques. Excess of intracellular calcium interferes with mitochondrial function, so we have an energy dysfunction, and interferes also with neurotransmission. Relevant and curiously is that all these processes, all these pathways that we know are in a different way related to each other. But now we have to understand where we want to go. Obviously, a much deeper discussion would be needed to explore the cascade potentially linked with the disease, and surely a lot is still to be discovered. Probably the key to further deepen the study of AD is, from one hand, how multiple pathways interact with each other toward the regulation of specific functions. On the other hand, I think it's important to include in the studies environmental factors, because we know that Alzheimer disease is a multifactorial disease. They can greatly influence the biological mechanism behind the AD, and in particular, I think it's really important, the epigenetic mechanism, as they are the way our genome responds, adapts to environmental change and stimuli. The last point is why what we want to know is genetic background. I think it's important because knowing it will permit us to evaluate the risk factor and the non-genetic measures to reduce the onset risk. And then knowing it will open to new potential targets for treatment. I think that we have to go toward the personalized treatment, personalized medicine, as we would be able to divide AD cases in subtypes according to their origin and treat each subtype in a specific way. Thank you for your attention and your time. For any questions or comments, you can send me an email to Crisafulli@unimed.it.